Hi guys, after seeing my lysosomal associated disease or lysosomal storage disease presentation, many people ask me to do why don't you do a presentation on organellar associated disease. So today we can discuss about organellar associated diseases. Organelles contribute to a disease in several ways. The first way is that an organelle itself becomes dysfunction because of the defective molecules present in it. Some enzymes or proteins present in the organelle may be defective, so that the organelle itself become defective, or because of the damage by the exposure of harmful substances. Some harmful substances such as chemical, heavy metal, free radicals such as exposure can result in the damaging of that organelle. The second way in which organelle will get damaged is through its normal function exacerbate damage occurring elsewhere in the cells. So, that is, for example, as we have seen, misfolded proteins in ER can trigger apoptosis even in the circumstances which it is counterproductive. That is, the organelle may be function, but something formed in the organelle can kill the entire cells. That is, in the case of ER, some misfolded proteins can result in the apoptosis of the cell. Coming to endoplasmic reticulum associated disease, the ER plays a central role in the synthesis of proteins and lipids. That any disturbance in its function can cause or result in serious consequences. The misfolded proteins coded by mutated genes and ER stress are the main causes of ER associated diseases. The first ER associated disease which we are discussing today is cystic fibrosis. Is a prominent example for the disease caused by misfolded proteins. Cystic fibrosis is ultimately a fatal inherited disorder in which a lack of a specific type of plasma membrane chloride channel, that is the cystic fibrosis transmembrane regulator, that is the CFTR. The lack of or deficiency of this chloride channel results in the accumulation of thick mucus in several organs, resulting such as lungs, pancreas, etc., resulting in the disease called cystic fibrosis. The misfolded CFTR proteins get trapped in the ER and subsequently degraded. So it results in the deficiency of CFTR proteins. So this results in the accumulation of mucus. So this results in cystic fibrosis disease. ER stress is another cause of disease associated with the endoplasmic reticulum, induced by a variety of conditions such as protein aggregation, CO2 plus depletion, glucose deprivation, and fatty acid overload can result in severe cell dysfunction and death. Because of this ER stress, these common diseases can occur. Alzheimer's, Huntington's, Parkinson's, even in heart diseases, diabetes is associated with ER stress. ER stress can result in protein aggregation, CO2 plus depletion, glucose deprivation, and fatty acid overload, which can result in these serious diseases. GOLGI associated diseases. The most common recognized GOLGI linked diseases are the 15 congenital disorders of glycosylation. That is, glycosylation is the covalent linkage of carbohydrate group to the polypeptide or lipid molecule. The deficiency of these glycosylation proteins or enzymes, glycosylation linked transport proteins, result in this serious disease such as CDGs. The CDG can result in the death of the individual at the age of 2. The symptoms include mental retardation, seizures, and liver diseases. Nuclear membrane associated diseases. Many diseases are attributed to the defects in nuclear envelope, which occur in the genes that code for lamin, a cytoskeleton components of the nuclear lamina, and emerin, an inner membrane protein. These two proteins, the mutations in these two protein genes result in lamin and emerin, result in most of the nuclear envelope associated diseases. Progeria, a fatal childhood disease, is characterized by the premature aging of musculoskeletal and cardiovascular system, is linked to a specific mutation in laminae genes, which forms the nuclear lamina. One other forms of disease is the Emery diffuse muscular dystrophy, is caused by the absence of or mutation to the genes that code for emerin. So laminin and emerin gene mutation can result in progeria as well as emery diffuse muscular dystrophy respectively. The cellular components of the nuclear envelope deficit include fragile nuclear membrane, altered regulation of DNA replication and transcription, low tolerance of mechanical stress, etc. Next, we can discuss about the disease associated with vesicular organelle. That is a disease associated with the link to lysosome versus peroxisomes, which we have detailedly discussed in the earlier slide, but we can break it down. Lysosomal storage diseases is a group of genetic disorders caused by the absence of one or more lysosome enzyme, resulting in the accumulation of undigested material within the lysosome. These type of diseases include Tay-Sachs disease, Gaucher's disease, and Pomme's disease. Another disease associated with lysosome is ISL disease. ISL disease which involved in the deficiency or problem in importing the lysosomal enzyme into the lysosome, so that the targeting of the lysosomal enzyme to the lysosome has a defect, so that lysosomal proteins will accumulate in the cytosol. That is the ISL disease. And these disease symptoms include mental retardation, heart disease, and respiratory failure. Plasma membrane plays an important role in wide diversity of diseases. Diseases such as cystic fibrosis, diabetes, familial hypercholesteremia is directly caused by the defective or missing membrane proteins. And several diseases which are caused by microorganisms such as Listeria, Salmonella, Shigella, HIV. Most of these organisms come in contact with the receptors present in the cell membrane, so thereby gaining entrance to the organism or the cell. So plasma membrane is the main site in which the infection starts, and as well as some other diseases in which bacteria will release some toxins to injure the cells. Once the toxin is bound to the plasma membrane receptor, a pore is formed and toxin is taken in, and which can result in damage to the cells. That type of disease which include cholera, pertussis, whooping cough, and diphtheria toxins. Mainly the HIV, Listeria, Salmonella, Shigella, etc. binds to the plasma membrane receptor and gain entry to the cell by endocytosis. But in the case of cholera, pertussis, as well as diphtheria, they produce a toxin which binds to the plasma membrane and taken in, and which can cause damage to the cells. These two types of way of infection caused by these microorganisms, as well as cystic fibrosis, diabetes, and hypercholesteremia is also associated with the deficiency of various proteins present in the plasma membrane. Thank you for viewing this video. Please subscribe to this channel. I hope you are enjoying this my video. You can it will be helpful for you to preparing most of the competitive exam. Hope you subscribe the channel. Thank you for viewing this video. Thank you.